Chapter 8 deals with recombinant DNA technology. So we're going to start looking at how do we apply some of these concepts that we have learned. Biotechnology is using microorganisms to make some type of a product that we want. Recombinant DNA technology, DNA is deoxyribonucleic acid, remember. This is intentionally modifying the DNA, maybe adding a particular gene to have organisms make something for us. There's 3 different goals. Number 1, possibly to eliminate some phenotypic trait that is undesirable. Remember, phenotypic traits are those that you actually see. How are they determined? By what's on the DNA. So you might have to remove the undesirable gene and replace it with the proper gene or the one that you want. Another goal is possibly to combine beneficial traits from other organisms into one. Or probably one of the most common things is to create an organism. It would be called a recombinant organism because it has DNA that was not naturally there, but it allows it to produce something that we need as humans. So an example of that would be, say, human insulin that's needed by diabetics. We have now taken the gene that is required for making the insulin and place that into bacteria and have the bacteria produce human insulin. So in a nutshell, this diagram is showing what we do with this recombinant DNA technology. In this example, it's showing the microorganism is going to be a bacteria and it has a plasmid. So you're going to isolate or remove that plasmid from the bacteria. Over on the right-hand side, you can see there's some DNA there. It has a gene that you're interested in. Who knows what it could be? Maybe for human growth hormone. It could be an insulin gene. Some of you may wish it would be a gene for allowing you to make A's on all of your microbiology exams. Whatever it is, it's a gene that you have determined you're interested in that. So the first step, isolate the plasmid. Remove it from the bacteria. Now, there are enzymes that are called restriction enzymes that will cut the DNA in certain spots. And so you're going to take the DNA that you're interested in, add these restriction enzymes, cut up that DNA so you can remove a piece of the DNA that you want. And then you're going to insert that DNA that you want into that plasmid. So great, now you put that DNA in the plasmid that you want, stick the plasmid that has a gene in it back into the bacteria. Now grow the bacteria and it will be producing whatever it is, the product is that you're interested from that gene. And like I say, you can use that for producing various enzymes, various antibiotics we do this for. It truly is just dependent totally on what that particular gene is. So in a nutshell, that's what we are doing. Mutagens, these are physical or chemical agents that will produce mutations. And we will often use that in a research setting to try to create changes in the DNA, which in turn will create changes in the phenotypes, what you physically see. So then you are trying to select for the best characteristics. Oftentimes we will use the enzyme reverse transcriptase to make what's known as complementary DNA. When you are talking about DNA from eukaryotic cells, they have introns in them that need to be removed before you can have protein synthesis from that. Prokaryotic cells don't know how to do that. So if you are going to take a eukaryotic gene and place it into a prokaryotic cell, what you really need to do is not place that original DNA in there. You need to place what's known as complementary DNA or cDNA in it. cDNA does not have the introns in it. How do you go about doing this? You take your eukaryotic cell, you allow it to go through and make its messenger RNA, still has the introns in it. Then it's going to go through that modification process of where it cuts the introns out, and you end up with that quote-unquote mature mRNA. You have that mRNA, it's got the cap on one side, the tail on the other, and you've already removed those introns and then spliced the remaining bits together. If you can use DNA as a template for RNA, then you can also use RNA as a template to make DNA. And that's how you're going to make the cDNA, is that you are going to use that mRNA that already has the introns cut out. Reverse transcriptase is the enzyme that is used. Where does it come from? It actually comes from a group of viruses called retroviruses that then use RNA as a template to make the DNA. And that's how you're going to make the cDNA. And then that has the introns removed, so it's a short little piece of the DNA. Now you can put that cDNA into the prokaryotic cells. Synthetic nucleic acids are simply DNA and RNA that have been produced artificially. You actually have machines where you can add the sugars and the phosphate, and you can add the base sequences in whatever order you wish. It's called a DNA synthesizer, and you just type in the order that you want, the adenines, thymines, guanines, and cytosines, and it will make it for you. It's been used in research for helping to determine genetic code, for creating specific genes for making particular proteins. Sometimes you make them as probes. So there's a lot of different uses for them. Restriction enzymes are those enzymes that will cut the DNA. They cut the DNA molecules at very specific sites depending on which restriction enzyme you use. There's hundreds of them. They will recognize a certain base sequence, usually anywhere from 4 to 6 bases, and it has to be in that order. If it sees it, then it will cut the DNA in a particular site. When it cuts it, it either cuts it with sticky ends or blunt ends. Blunt ends are what we see in figure D on the right-hand side. It's a straight cut. Both strands are cut in the same spot. So it's just like taking a knife, just sliced it right down. Sticky ends, it's a jagged cut, as you can see on figure A. So once again, it still has to recognize that certain base sequence. It will cut in certain areas. If you have a choice, a lot of people prefer, at least those that I'm personally familiar with, prefer using sticky ends, because when you go, what you're going to do is you're going to cut the DNA and then you're going to insert that gene of choice in there. And if you have that jagged edge, you just have a better chance of it lining up. And then to seal the edges together, you would use ligase. Remember DNA ligase? It's kind of like that DNA super glue. It seals the bonds so you don't have that gap where the cut was. Vectors, simply, how are you going to move the gene into a cell? Vectors are usually very small enough that you can manipulate it very easily in a lab. It will survive inside the cell. And it usually has some type of a marker on it so you can ensure or be able to detect, did you get the gene where you thought you got it? Oftentimes it's plasmids. So once again, this is showing just an example of how you're producing a recombinant vector. So you have a plasmid, you have your restriction enzyme, you add to it, you cut it. You use the same restriction enzyme to cut the DNA, the cDNA, so that way you have matching sticky ends. You insert the gene of choice in, you use the ligase to seal the edges, and then you put that recombinant plasmid back into the bacteria and then grow it and monitor that. You have little markers that you have to ensure that you can tell, yes, it was successful in this particular bacteria, maybe not in another. If you are going through the process of making these recombinant plasmids and inserting them into these recombinant, say in the examples we've seen, bacteria, you don't want to lose all that work. You do all that work for what? So you don't want to have to start all over again. So you maintain what we call a gene library. That's going to be basically a culture of whether it's bacteria or whether it's a virus that has it, so that you can always go back there to retrieve the DNA that you're interested in. So how do you produce it? Once again, you go through that whole process of isolating the genome. You may insert it into a vector. The vector is placed into multiple culture cells. Now, in this case, they're showing multiple genes, so it's not just one, multiple ones. You grow all those up and keep very good records of what was put into what organism, and then you grow those and maintain them so you can always go back to them later. So that's what you just call your gene library. So what happens if you have just a little bit of DNA, not a whole lot available for use? You can amplify the DNA. You can make more copies of the DNA using the polymerase chain reaction, or PCR. It is a way to amplify or increase the amount of DNA for whether it's for research purposes, etc., for use later. It is a repetitive process. It involves basically denaturing, priming, and then extending. And there's a process that's all automated now. And this shows how you go through those three processes of the denaturation, priming, and then extension. The bottom line is, you can see here, each time you run a cycle, you are increasing, you're doubling the amount of DNA that you have. This can be used for several different purposes, whether for research, whether for forensics. You don't need very much to increase it. You must be able to find, you go through this work, you must be able to find the clone that has, the organism that has that DNA in it that you're interested in. Because you've cut up the DNA, you're putting it in different bacteria, let's say. Well, all of them are getting different pieces. So you have to find out which one has the one that I want. You'll use a probe. You can also take the DNA and run what we call an electrophoresis gel. It's going to separate the DNA. What you do, you would take the DNA, you cut it up into those pieces, you run it on an electrophoresis gel. It's going to separate all those DNA pieces based on the size, the shape, and the electrical charge of it. You make this gel, its consistency almost kind of like Jell-O. You add your samples at one end of the gel, and then you hook it up to an electrical current. And it's going to flow from one end, usually the negative, down to the positive end. Because it's moving through this gel-like structure, the larger the size, the slower it's going to move. The smaller ones move faster. So you can get a relative idea of size and comparisons by looking at this. So you can see in photo A the apparatus of how you would set it up. You have those different wells. As you can see, they're labeled in this one A through E. So you can put your control is in E, so that has known sizes. So as the fragments will separate, once you have turned the charge on, the electrical current on, you'll see those numbers in parentheses in column E are showing the different size of the fragments. So the smaller ones move fastest. And then your samples that are placed in the other wells, as you can see, you get different banding patterns, and you can line them up to get a relative idea of how large each of those fragments are. Oh, this one matches up right with 15. Okay, then this fragment must be about 15 kilobase pairs. And then you can see in the photo on B how it actually looks once you have run it. And oftentimes there are dyes that are placed in there, and it's easier to put it on a base and you light it up to be able to see the banding pattern. When you are separating out DNA fragments, that's referred to as a southern blot. So with that southern blot, you're using the DNA. From the gel, you transfer it to what's known as a nitrocellulose membrane, and then you can add probes that will match if it's compatible base pairing. So you can see, oh, okay, it's this fragment at 15 that I'm interested in. I don't care about the other ones. A northern blot, you may hear of that sometimes. It's very similar, except that you're using RNA. So southern blots with DNA, northern blots RNA. And just so you know, there is a western blot, which is protein. DNA microarrays, these consist of molecules of single-stranded DNA. Usually you will use a fluorescent dye, fluorescent dye that is attached to DNA, it's washed over. It's only going to adhere where there's that complementary base pairing. And it's a way of monitoring sometimes gene expression. You can use this for diagnosis for infection, and you can also use it for identification of certain microorganisms, especially if you're using environmental samples where it's sometimes hard to isolate from. So this is just showing in this diagram the use of a DNA micro array where, once again, the DNA is labeled. It's got this fluorescent dye that's attached to it. The whole goal of your recombinant DNA technology is to insert DNA from one source into a cell. Now, we've already discussed there are some natural methods that this can happen: transformation, transduction, and conjugation with bacteria. There are also some artificial means or processes of doing this: electroporation, protoplast fusion, and injection. So protoplast fusion and electroporation are shown here, where basically electroporation, you have your bacteria, you apply an electrical field, and it creates pores or openings in the cell wall and cell membrane that then allows the DNA from another source to be able to move in. And yes, the cell's going to repair and make new components of cell membrane and cell wall to repair those pores or those holes. In protoplast fusion, what happens here is you remove the cell wall, leaving just the cell membrane. And then polyethylene glycol is a type of detergent, basically, that will then allow the two cells to fuse together. The DNA combines. Now, the cell will eventually synthesize a new cell wall. Using injection methods, you can use either C is showing with a gene gun. You actually have these special type of, it's like a 22 caliber shell. You have your projectile, and on the end of it you coat beads with DNA, and you actually fire this and shoot it into the cell. So you're literally shooting DNA into the recipient cell. When I was at University of Idaho, the biology department did obtain one of these, which was kind of a strange thing. They were on the cutting edge there, but to see all the signs, gun in use in lab, caution. It's like, well, what are you guys doing? Oh, we're just shooting DNA into the cells. Microinjection is when you're using a micropipette. You can drop very small amounts of DNA, and literally, as you can see in this diagram, it takes some skill and technique, learned techniques, to be able literally just to inject in the DNA into the cell using a micropipette. It's like taking a needle and injecting it in. Gene mapping is simply trying to figure out where on the nucleic acid, the entire molecule, where the gene's located. It can give you ideas about characteristics of the organisms, how different genes may be related to one another, and things such as metabolism. Initially, it was very hard in the early days of this field. There's universal methods used now. It's much easier to do. since they found restriction enzymes, and they often use what's known as fluorescent in situ hybridization. So as you can see, they use fluorescent dyes, gives you an idea where various things are located, various genes. In terms of nucleotide sequencing, this is just basically taking the DNA and figuring out what is the base sequence. Is it G, A, T, T, C, G, G, etc. And they just list them out, gives you an idea of where the genes are located, what the base sequence are for the various genes. And so they have been able to determine the DNA sequence of many organisms, including humans. And now they're moving into what we refer to as the next generation DNA sequencing, allowing us to make complementary base strands, etc. If you know what is normal, then you can see what is abnormal and hopefully be able to fix if there is a disease or disorder. Functional genomics, using basically genomics to determine what the gene products do. So great, you know that here's a base sequence. This is for gene A, but what does gene A do? And so along with this whole field of genetic mapping is trying to figure out where the genes are and what are the products. This is an example of some of the different tools or techniques that we have, what the potential applications of them are. Most microorganisms have never been grown in the lab. We often know they are present in the environment because of the DNA. We can do maybe add probes, add fluorescent dyes to soil, and we can see the DNA there, but we cannot grow the organisms. They don't come with a manual of, hey, this is what I need to grow. So from the DNA fingerprints, the DNA copies, we've been able to identify a lot of species. As an example, over 500 species of bacteria have been identified that were isolated just from the mouths of humans. The idea is ultimately we're at the stage we're wanting to get an idea of how many different species and what are those species that are found in what we refer to as the microbiome. The environment, whether it's, say, on your skin, in your mouth, in your intestines, in a soil environment. What are the microorganisms that are living there? There's a lot we don't know. In terms of some applications for recombinant DNA technology, certainly we have used this for making different proteins. We have used this for making vaccines. The advantage of using recombinant DNA for vaccines is you don't have to use the entire virus or the entire bacteria. You just make what we call subunit vaccines. It's just components of typically proteins that are on the cell surface of whatever the microorganism is that then can be used to stimulate your immune response. If you don't have the entire organism, there's no way you're going to get sick from it. We have been able to use this to help introduce basically the whole field of pest management in agriculture, introduce genes into certain plants and organisms that will act to help ideally reduce the use of, say, chemical pest management practices. For genetic screening, we've been able to use this, helps to identify different DNA from, say, viruses and tissue samples, be able to see if individuals maybe have genetic screening, do they have a mutation that puts them at higher risks for developing another disorder or disease. And then the idea also is to ultimately get to gene therapy where you can replace a missing or a defective gene with a normal copy. Can use this for medical diagnosis, for helping with transplants. Animals are often used initially as study, and then we see if we can progress this to humans. Agricultural applications, this has been going on for a long, long time, probably longer than most people realize, of using recombinant plants and animals. You add different genes. So just because you add a new gene that wasn't there, that now makes it recombinant. This is also what is known as genetically modified because you added a gene in there that is not found in the wild type, and that's referred to as GMOs. In terms of agriculture with plants, this is often done to increase the nutritional value of certain plants. This has been done also, as said, for herbicide tolerance. You can add it into certain genes into, say, your crop plants. Add a gene that confers resistance to certain chemicals, so now you can spray the herbicide. It kills the weed, but not your plant. Also, salt tolerance is another thing that they are looking at, so that you may be able to grow plants in a high salt environment without killing the plant. Freeze tolerance, I actually knew the woman who developed this, where it was the first release of a genetically modified organism into the environment. It was in California on strawberry plants. It was referred to as the ice minus mutant, and what it does, it's a bacteria. It is non-pathogenic, and they add it to a solution that they can spray. They were looking mostly in the springtime when you have a late frost. And what you do is you take this organism, and it's referred to, as I said, the ice minus mutant. And when you grow it in a liquid solution, you spray that liquid solution out. Instead of freezing at 32 degrees Fahrenheit, it does not freeze until 28 degrees Fahrenheit, which you might say, well, that's not that big of a difference. Well, that slight difference in late spring, when you have a late frost, it can mean the difference between losing your whole crop of strawberries or not. And so this, like I said, I knew the woman that helped develop this. It was the first release of a GMO. It was very successful, like I say, in helping to prevent the crops from freezing with a late frost. Another thing is you can add a toxin that's produced by bacteria. It's a natural toxin to insects. It does not have an adverse effect on humans. Once again, instead of spraying chemicals, it's a more natural, organic way of pest management. This just shows a picture of some genetically modified papaya plants. These are just some additional examples of, from agriculture. Like I say, this has been going on for a long time. Whether it's bovine growth hormone added to cattle to have them gain weight more rapidly, the meat has a lower fat content, they produce more milk. Adding beta carotene into rice. Beta carotene is needed to help produce vitamin A, which is necessary for proper vision. And the reason this has been added to rice is that rice is used so often as a stable food source for underdeveloped and poor countries. And so if you can add this to the rice naturally, this gene, so the rice is producing not only the rice as a food source, but also now a component, it's a vitamin precursor that is necessary for proper health. Long-term effects are not known. Here comes the big question in science, regardless of the field, is how long is long-term? So far, they have not seen adverse effects from the use of GMOs. There is concern is could there be natural genetic transfer from these transgenic or recombinant organisms into the wild type? Usually there are certain safety mechanisms that are put in so that that cannot occur. At this point in time, we do not know what the long-term effects may be, but we have not seen adverse effects. There are very strict standards that are imposed on the labs in terms of regulating using recombinant DNA. Scientists are pretty good about regulating themselves, but the concern has always been, You can use this for good, but you can also use it in harmful ways. Potentially could use this to create biological weapons, and that is what the concern is. So there are very strict regulatory components added to any type of recombinant DNA technology research and practices. There are ethical issues. For every question you ask, there are more questions. Um, just because you have the technology to do something does not necessarily mean you should do it. And what I may consider to be okay, someone else may not. And so it does bring up additional questions in science that you have to look at. A lot of these are ethical questions. If you start looking at altering the genes, do you do this with plants? Is that okay? Can you do this with animals? Is that okay? Can you do this with humans? Is that okay or not? Who would pay for it? What are the privacy rights? Do you do this with routine screenings? Um, Years ago, when I was doing research, there came a question. Somebody was working with a recombinant organism, and the question came up of, you cannot patent an organism. You can't go out and patent E. coli. You cannot patent a corn plant. You cannot patent an oak tree. They exist naturally in the wild. But what happens if you alter them genetically? They're not the same. So if you did that in the lab and created this new strain in the lab, can you patent that? Can you get profits from it? Who has the rights to that? So there's a lot of questions that come up, even ethical questions in terms of, you can say, well, this might sound great, that you could, for different disorders, different abnormalities, different diseases, can you correct this using gene therapy? Do you have a right to do that? Who determines what is normal and what's abnormal? So there are a lot of ethical questions to ask yourself, to ask if you're involved with this.